talking about 4.1.1.3, and it's talking about cell specialization. We're going to look at the structure and the function and how structure relates to function. So everything with structure always relates back to function. So that's the important thing. In Key Stage 3, you might have learned about tissues, organs, organ systems, and what whole organisms are. But it is key to know the definitions between them. Today's video we won't go specifically on these, but I'll give you a brief overview of what these are. So different sorts of cells will make up tissue. And then similar sort of cells will make up some tissue. Different sorts of tissue will make up an organ, and different organs will make a different an organ system, and the different organ systems will make a whole organism. Okay, so you can have muscle tissue, which is made up of actin and myosin cells. You can have tissue cells, skin tissue, which is made out of something called keratinocytes, which are basically skin cells. So therefore and so forth. I think there is another specification point where you go into more detail about these in another one. But for now, this is the main area of our concern today. Cells may be specialized to carry out a specific function, and they've noted down sperm cells, nerve cells, muscle cells in animals, and they've noted down root hair cells, xylem, and phloem in plants. And we'll go through what each of these do. So let's get through those. 4.1.1.3 cell specialization. The first thing to know is how the following cells that we're going to talk about structured towards their function. Okay, so everything that has a structure will have a function towards it, so it's there for a reason. It's not just there for no reason. So we'll show it first with the sperm cell. So in the exam they may give you a diagram and you're supposed to interpret it from the diagram, or what they might do is give you some other cell and they'll tell you a little bit about that cell. So the ones in the specification are not limited to what they'll ask. They can ask about something else, but you have to interpret it in your own way to see how is this structure going to help this cell do its job. So we're going to go through the ones they've talked about in the specification, but you need to be able to apply this knowledge into other scenarios as well. So you don't be surprised if they talk about egg cells or something else that is within another part of your specification, but they will ask you about how is this structure affecting its function. Remember, that's key for the exam. So don't just be like, I need to learn what sperm cell does. No, you need to be able to critically think, how does your structure relate to its function? So let's go for sperm cells. So sperm cells have a tail. The tail is there to help it move, and the only way it's going to move, it doesn't move just by nothing. It needs energy. And where does energy come from? The mitochondria. Again, many mitochondrion make mitochondria. This is just a singular version. So there's a lot of mitochondria which is providing a lot of energy, and in biology energy is ATP, right? So it provides a lot of this energy. And the head region here, when you have an egg, okay, you have an egg, this has to come through and break the outer shell. Okay, to break that outer shell you need acrosomes, which are enzymes that break it down and help it to penetrate through into the egg. Then we have nerve cells. So nerve cells have these myelin sheath. That's the most important thing to remember, and this is made up of Schwann cells. Okay, and Schwann cells are in your peripheral nervous system, so everything with your hands and everything. Those are what you're going to need to know for Schwann cells. That's all you're going to need to know. There is another type of cell that makes myelin sheath called the oligodendrocytes. However, these are residing in your your central nervous system, but they are unlikely to ask about this unless it's an application question where they'll give you a little bit more detail about what these are. But they have a similar role to Schwann cells, so just remember they make myelin sheath as well. And what this myelin sheath does is allows for faster conduction of electricity. Why does this allow for faster conduction? Well, these myelin sheath are made up of sort of lipid molecules. So essentially your electric current, so let's think of these as your lipid molecules. Your electric current can't now go through and just go anywhere it wants, right? It can't just go out. It can't do that. So what will happen is the electric current will actually do this, go up, down, up, down, up, down. So stay within its place because it's in that myelin sheath. And then it'll go through much faster than if it went all the way out for some reason and then came back and then went like this. That's a longer direction. It's much easier for them to stay in that area rather than just sparking out. If someone has damage in this area, that can cause problems as well. Something called multiple sclerosis, where some people have damage in these areas, and what can happen is people can start spasming into different parts of their muscles. So that's just to be aware about the nerve cells, and why this myelin sheath is so important. Later on, you're going to learn more about, in your A levels, you can learn about why there's these gaps as well, but I won't go into detail about this here. Muscle cells in animals. There's something called actin and myosin cells that make these strong contractions. These are the sort of cells that make it up. So you will have something like this, which is a myosin head, and you have a actin attachment point. So this myosin can attach onto this actin point, and it allows it to contract. Your specification has it specified as to muscle cells in this one. When we come towards, if there is a muscle specification point, we'll go into detail, but all you need to know is there's actin and myosin that help to make these strong contractions. Remember, muscles are used for movement, so you're going to need mitochondria. If there's anything to do with movement, mitochondria is always a good safe option to put in the exam, and it's used for energy. And it's also constantly renewed, so it's not used up one time and that's it. It's constantly renewed and can be used again and again, the actin and myosin. Moving on to plant cells, we have the root hair cells. So with plant cells, you can always talk about the cellulose that you can see here. So the cellulose obviously prevents cells from lysing, so from lysis. So lysis just means it just won't break, like it just won't explode suddenly because there's a ton of water coming in. Okay, so it won't do that. So cellulose allows that cell to survive in that really high water environment. So if there's too much water here, it won't just break down. Also, what we have is a large surface area. So essentially, these are root hairs and they'll have smaller hairs as well, which essentially allows for a lot more water to come in because of the large surface area. So this is the key point they would want you to make in the exam. They also do have channel transporters as well in here, because they need to take in mineral ions, and usually the mineral ions aren't within the concentration gradient. Because they're going against the concentration gradient, we need to use active transport. Okay, so if there is more ions outside than inside, that's fine, so you can just go in. However, what usually tends to be the case is there might be only a few ions outside and a lot inside the cell, so you need to use active transport. You need to use ATP, and you need to use active transport. ATP is just the energy. Use active transport to move in that mineral ion. Now we come into xylem and phloem cells. So xylem is the one carrying water, and it's waterproof because it is lignified. So that means all the cells are dead essentially. So that's a key difference between xylem and phloem cells. Xylem cells are quite pretty much dead, and they move usually in one way. The lignification is important for waterproofing and strengthening of that vessel, and it usually allows water through. There might be other minerals, but it's usually water. Phloem cells have sieve tubes. So can you see here? There's like little sieves. So you can see in this one, like there's like little sieves which allows water through. And it also has companion cells, which transport substance in the phloem, and it requires energy. So essentially, these companion cells will put in substance into this. When we look at things like sucrose loading and translocation, this becomes really important. So cells here will then dump certain minerals and sucrose into this phloem, which can then go up and down depending on where it needs to go in the body. So looking back at the specification, we have covered what the structure to the function is, and we've covered about all these cells, so we're done here. Now let's talk about the different things you can look at to gain extra knowledge or something you want to learn more about. So the first thing you can learn more about is, if you learned about sperm cells, look about the egg cells. What are the differences? You can look there. Another thing you could look more into is, how does this sperm cell actually interact with this egg cell? So there's something called the zona pellucida you can look more in depth about. So you can look more about this zona pellucida and how does the sperm cell and the zona pellucida actually interact. There is actually receptors within called isomo and Juno, so you can look into these receptors as well. So this is more advanced things, but you can look into them if you wish for your own knowledge. Nerve cells, you can look into something called saltatory conduction. That's the thing I was mentioning. Conduction, I think that's how you spell it. Look into saltatory conduction. That's where it essentially skips and makes the nerve conduction much faster as well. So the other thing you can look into is, how do muscle cells actually work. Another thing you can look into in the root hair cells is looking into particularly how do these channel transporters work and the different types of active transport. So you can have primary and secondary. So these are just extra things you can look into if you wish to. So I'm just giving you the directions to where you should look. Xylem and phloem, the things you can look into is how are different species got different types of xylem and phloem. That's just extra things you can look into. But as I said, these are not going to be examined. These are just extra things you can look into to just broaden your knowledge.